Welcome to the FENS Friday webinar organized by the FENS Committee for Higher Education and Training from FENS. The topic of webinar today is translational electrophysiological assessment of brain excitability. And this topic was selected in a call for ideas organized by FENS Chat Committee. And in this call, you can all send your suggestions for training or educational activities. So just for short, FENS Committee for Higher Education and Training aims to actively support continued higher education and training in neuroscience for early career scientists. You can also check the FENS website, fens.org/training for more information. Today, it's my pleasure to introduce the moderator of this session, Ioana Mândruță, which is an associate professor at Neurology Department, University Hospital of Bucharest. She is the head of Epilepsy Monitoring Unit, National Coordinator of Drug-Resistant Epilepsy Program. Ioana is interested in pre-surgical evaluation, intracranial recordings, and brain stimulation in drug-resistant epilepsy. Ioana, you have the screen.
Hello, everybody. It is my great pleasure today to moderate this webinar. Of course, assessing brain excitability is a critical aspect of understanding brain functions and diagnosing various neurological conditions. Several neurophysiological techniques are developed in the last decades, each of them providing unique insights into the excitatory and inhibitory processes within the brain. This FENS Friday webinar will show some of these methods and discuss the application in human pathological states. Our speakers today will be well-known researchers in the field of neurophysiology. And I start with the first speaker, who is Dr. Nicolas Gaspard. He is the head of the Department of Neurology at the University Hospital in Brussels and professor of neurology at Université Libre de Bruxelles in Belgium. He is also an adjunct professor in the Department of Neurology in Yale Medical School in US. Since 2023, he has served as the chair of the Belgian Clinical Neurophysiology Society. Professor Gaspard's research interest is in electrophysiology and signal analysis in ICU, status epilepticus, and epilepsy. His title, his presentation will be the phenomenology and clinical use of EEG reactivity in men. The second speaker will be Dr. Mirela Simon, who earned her medical degree from Carol Davila University of Medicine and Pharmacy in Bucharest, Romania, and later a Master of Science degree from the Harvard School of Public Health in Boston, USA. Currently, she serves as an associate professor of neurology at Harvard Medical School and Massachusetts Grant Hospital and Brigham and Women's Hospital. Dr. Simon's primary research focus centers on enhancing and developing innovative techniques for neurophysiologic mapping of the central nervous system. Her presentation topic will be variability of cortical excitability in intraoperative neurophysiology. The third speaker is Mihai Moldovan, who is associate professor and past president of the National Neuroscience Society of Romania, graduated from Carol Davila University of Medicine and Pharmacy in Bucharest, Romania. He earned his PhD in neuroscience from the University of Copenhagen, where he continued his academic and clinical career at Rigshospitalet in Neurology Department. His ongoing research and innovation activity in Copenhagen as well as in Bucharest focuses on developing translational neurophysiological methods for functional studies of the peripheral and central nervous systems. He is particularly interested in the assessment of the excitability at the axonal level, as well as at the neural network level, with broad diagnostic and therapeutic implications for neurological diseases. His title of presentation will be very interesting: to burst or not to burst, an EEG story. The last but not least will be Irina Wane. Irina Wane is a young neurology specialist and a scientific researcher who is part of my group in Bucharest in the epilepsy monitoring unit and neurology department at University Emergency Hospital in Bucharest, Romania. She completed her PhD thesis approaching the functional mapping and connectivity of the cingulate gyrus. Dr. Wane's research focus on the invasive presurgical exploration of patients with drug-resistant epilepsy, in particular functional brain mapping and brain connectivity using electrical cortical stimulation during stereo EEG. Her title will be Direct Electrical Stimulation with Intracranial Electrodes for the Study of Cortical Excitability and Functional Connectivity. And now the rules of our webinar will be that the questions will be answered at the end of the 4 talks during the Q&A session. Simply write in the Q&A session if you have questions during the talks, and you will find the button at the bottom of the Zoom window where the chat button is disabled. So please use the Q&A section to address your questions. And keep in mind, anonymous questions are not allowed. So we should start with the first speaker. So please, Professor Gaspar.
There you go. Sorry about that. Thank you very much for having me today. It's a pleasure to be online with the other panelists to contribute to this webinar. I'm mostly a clinical neurologist and clinical neurophysiologist. So what I'm going to try to do in the next 10 minutes is to give you a broader overview of what is EEG reactivity, what we call EEG reactivity in human patients, and how it is helpful or how it could be helpful in several clinical situations. The concept of EEG reactivity is really tied to the discovery of the ascending reticular arousal system. And I think one of the seminal work on that is the work by Maroudi and Magoon, who had this model of encéphale isolé, which basically means disconnecting the entire brain from the rest of the nervous system in cats, and then stimulating different brain areas, especially in the brainstem. And in those experiments, what they were able to show is that stimulation of structures in the upper brainstem, nuclei that later were found to be, for instance, the locus coeruleus, the pediculopontine nucleus. Stimulating those regions would induce broad changes in the electrical activity of the cortex. This is electrocorticography, and during stimulation of the brainstem, of these regions in the brainstem, in the ascending reticular arousal system, stimulation of those regions leads to profound changes in cortical activity. So not only cortical activity, but also thalamic activity. And this, of course, was found later to be related to the fact that those structures in the upper brainstem were densely connected to non-specific nuclei in the thalamus, and the cortex were able to influence the activity of the entire thalamocortical assembly. And these are the areas that they identified as being able to induce those changes when stimulated, to the dark gray areas there, and all the other light gray areas were actually unable to lead to those changes. And interestingly, If you make the jump from cats to humans, and this is an interesting study done by Provisi and Damasio 20 years ago, using statistical lesion mapping. So taking MRIs from many patients with the same problem, being in a persistent coma, and looking at which brain regions were systematically damaged in those patients. They actually found that these were the exact same regions that Maruzzi and Magoon had identified as being able to induce activation in thalamocortical structures if they were stimulated. So Maruzzi and Magoon did a gain of function experiment, and Provisi and Damasio took advantage of a natural loss of function experiment to both demonstrate that these regions of the ascending reticular arousal system in the upper brainstem, in the midbrain and the upper pons, were really critical to turn on cortical and thalamocortical activity. And thalamocortical activity is, of course, really the key player in the generation of the EEG signal. Whether this EEG signal is normal, this is a normal posterior dominant rhythm, for instance, of an awake adult individuals. These are K complexes, sleep spindles, slow waves of sleep. So these are very normal brain activity that you can record with EEG, and they are generated by the interplay between the cortex and the thalamus. There's also indication that abnormal brain activity, abnormal cortical activity that you can record with the EEG in critically ill comatose patient, like generalized periodic discharge or burst, are also generated by dysfunctional thalamocortical interaction. So all cortical activity that you can record with EEG is generated thanks to these thalamocortical interplay and cortical interplay as well. So testing EEG reactivity for us clinical neurologists could be of tremendous interest, because it's a very simple way. It can be done at the bedside just by making noise, shaking the patient. I'll show you how it's done in a moment. So in a very simple way, it's an easy way to try to excite the brain and see what it is able to generate in terms of activity. And of course, the idea is that you don't see any EEG reactivity, it suggests that those key structures like the upper brainstem and the thalamocortical loops are injured to the point that recovery of consciousness is very unlikely. That being said, and despite decades of clinical work, if you look at the recent recommendations, for instance, of the European Resuscitation Council and European Society of Intensive Care Medicine for prognostication of awakening in patients who suffered anoxic brain injury, the recommendations actually are against using EEG reactivity to predict awakening or lack of awakening. And the reason, and this is nicely summarized in a systematic review done by Marie-Hélène Admiral a few years ago, the reason is that there are no standardized way of doing it. No standardized protocol to assess reactivity. It can be done in many different ways, and many groups do it in different ways. So there is no replicable standardized protocol, and there is also no standardized definition, or at least there was no standardized definition of what is EEG reactivity. So the suggestion after this systematic review was that we needed a precise, standardized simulation protocol and a standardized definition of reactivity. We have moved forward since then, and we did indeed work on a standardized definition of what is EEG reactivity, and this can be found in the standardized terminology from the American Clinical Neurophysiology Society that define reactivity as any change in cerebral EEG activity upon stimulation. And this may include changes in amplitude, in frequency, including attenuation of activity that was present before. And the suggestion is to classify the EEG as reactive, unreactive, unclear, or abnormal reactivity, which is those stimulus-induced rhythmic, periodic, or ictal discharges. Another progress that we've made in the recent years is to develop a standardized protocol for reactivity testing. This is work from our group, from Sarah Carroyer, where we actually tested a sequence of stimulation from very mild auditory to quite noxious stimulation. So mild auditory stimulation, that's calling the patient name, low auditory stimulation, that's actually yelling the patient name, clapping in hands, shaking the patient, tickling the nostril, squeezing the trapezius muscle. That's actually quite noxious, but there is little chance that you could injure the patient doing that. Suctioning the tube. These patients are often under mechanical ventilation, so they have an endotracheal tube you can suction. That's quite noxious. And finally, passive eye opening. And we compare this entire sequence to what we call the simplified sequence that only consisted in some noise, shaking, and trapezius squeezing. The idea is we wanted to have something that could be very simple and easily used at the bedside. And we actually found that there was fair inter-rater agreement between different EEG raters looking at the EEG upon this type of testing, and this was reasonably associated with the chance of waking up of post-anoxic coma. So now we have a standardized definition, and we have a standardized way of testing reactivity. But there are other problems. And this is something we did a few years ago now, looking at how much change there was upon reactivity. So far, I've presented you reactivity as a all or none phenomenon. There is no reactivity, but what if there were some reactivity, right? That the EEG could, in some patients, show massive changes, and in some patients, show no changes, and in some patients, show some changes, some in-between changes. And this is something we did a few years ago, basically looking at the difference in the spectral content of the EEG signal before and after stimulation. So this is before, this is after, this is quantitative EEG of this EEG segment. So this is in blue before stimulation, in red after stimulation. So already with the naked eye, you can see that there is a difference, and this difference is quite striking. If you quantify this, you actually find a probability that the EEG is reactive of 0.93, and this goes from 0 to 1. So this is an example of an EEG that's clearly very reactive. And the opposite is this EEG, where there is basically no change with stimulation and a very low probability. So interestingly, if we looked at 100 of these EEG clips, and what we found is that there were patients in the top left of this graph that had a very high probability of reactivity. And interestingly, when we compared the probability to what the human raters had said, we found that these cases were found by at least 2 of the 3 readers to clearly be reactive. And the opposite is also seen. So in these red boxes, you have patients where the algorithm says very low probability of reactivity. And again, at least 2 of the 3 raters had said that the EEG was not reactivated. But then you have this yellow bar of patients where actually it wasn't clear. Either all 3 raters said something different, or all of them said, no, that's unclear. We can't say if there is reactivity. Interestingly, the algorithm in those cases say provide a probability which is in between. So the bottom line is that the EEG reactivity is not a binary, a yes or no phenomenon. It's a spectrum of possible changes. And these changes include appearance of slow activity, desynchronization of the EEG, appearance of periodic discharge, stimulus-induced periodic discharge, or even stimulus-induced burst of activity on an otherwise flat background. So it's also not about only how much reactivity, it's also about what type of reactivity is seen. So the key message is that the EEG reactivity stimulation probably indicate preservation of the ascending reticular arousal system and some thalamocortical connectivity, basically preservation of the key structure that support consciousness. And so the presence of EEG reactivity should be seen as a sign of good neurological prognosis. It's mostly true, but the positive predictive value is clearly far from being 100%. And on the other end, absence of reactivity could be seen as a sign of poor neurological prognosis. That's often true, but again, the false positive rate is clearly higher than 0%. So it's not perfect yet. Can we use reactive testing in human comatose patients to make prediction? Yes, but there are still issues, and the main issue is to quantify, I think, the reactivity, to determine which type of activity is brought out by the stimulation, and also to quantify how much a change it represents from before stimulation. And that, of course, sets the stage for future works, including what Dr. Moldovan will tell you about in a minute. So that was my 10-minute introduction of a way to test brain excitability in comatose patients, which is EEG reactivity. Thank you very much for your attention.
Thank you very much for this very insightful presentation. And I will move now to the second speaker, Dr. Mirela Simon, about variability of cortical excitability in intraoperative neurophysiology.
Thank you very much, Jona, for the introduction. Good morning, good afternoon, everyone. It's a pleasure to be here, and I'd like to thank the organizers for inviting me to participate in this webinar. So in the next 10 minutes or so, I will give a brief overview from a clinician perspective on how variability of cortical excitability impacts what we do in the operating room, in particular when we map eloquent cortex in supratentorial surgery, such as brain tumor or epilepsy surgery. It's not moving forward. 1 second. I'm not sure why it's not moving. It's not advancing. Okay. These are my disclosures. So one of the challenges we face in the operating room is that we're dealing with a multifactorial environment. So there are many factors that can impact cortical excitability. And this is very important because we are always going to rely on thresholds for excitation or inhibition of eloquent cortex when we perform mapping procedures. So this is just a short list with the main factors that impact our stimulation thresholds. Anesthetics is a big part of it, either by type as well as by dose. Pathology, age, with just thinking of the fact that immature cortex is difficult to map. Sometimes we are not able to reach the depolarization threshold of M1 in children under general anesthesia. Temperature, blood flow, and last but not least, primary versus secondary functional cortical regions have different thresholds, with, in general, primary eloquent cortex having lower threshold than secondary M1 versus M2. Also, there are interlobar differences in this excitability and indirectly in thresholds, with temporal language areas having higher thresholds than anterior language areas such as Broca. So we try to look and see if we can identify factors that do impact our threshold, first in language mapping. And we wanted to see whether the proximity of the lesion to the map cortex, as well as the lobar location of the map cortex, have an impact on these thresholds. And sure enough, we use a multivariate regression analysis, linear regression analysis. And in this analysis, lesions that were in close proximity to the map cortex resulted in increase in our mapping threshold, as well as location of the map cortex in the temporal and parietal regions had higher thresholds when compared to anterior language areas such as Broca. And this is also can be seen here with highest thresholds in language mapping in patients in whom we map Wernicke and who had lesions in close proximity to the map cortex versus patients in whom we map Broca with lesions away from the map cortex. We did a similar analysis, but this time to look at factors that impact thresholds for mapping M1, both in awake and anesthetized patients, with the big culprits being, of course, general anesthesia. And we saw that patients who were anesthetized with inhalational agents had the highest thresholds, followed by patients under TIVA. And of course, the lowest threshold was in awake patient. All patients also, the depth of EEG suppression is appreciated by the average length flats in the burst suppression pattern. Seem to correlate well with the thresholds. With longer the flats, the deeper the suppression, the higher the thresholds. In a univariate analysis, all these factors, including older age, did have a significant impact on M1 thresholds. However, when we brought them together in a multivariate analysis, older age and the use of total intravenous anesthesia lost their significance because we already adjusted for the depth of suppression, knowing that, older brains tend to go faster in the suppression. And once we corrected for the depth of suppression, the pure use of TIVA became insignificant, whereas the impact of inhalation agents was still significant. What we also notice when we map under general anesthesia motor cortex, there is really a wide range of thresholds that we are dealing with. And we did have patients who were anesthetized. We mapped it, 1.5 milliamps, and we had other patients, particularly children, in whom we reached 25 milliamps, the max that our machine can deliver, and we barely map, or some of them we couldn't map because the thresholds were higher. So it is thus important to know when to gauge, to try to estimate the threshold on an individual basis, because otherwise it will take a long time to start from like 1 milliamp and slowly go up to 25 milliamps. So understanding, the level of cortical excitability, if you like, in that particular patient is very important. But we also have the reverse. So we do have patients who present, for example, with seizures, particularly brain tumor patients. We notice that they tend to seize frequently even under general anesthesia when we perform motor mapping. And we are looking for biomarkers to detect whether there is an increased perilandic network excitability, because in this case, we are not going to start stimulating at 8-9 milliamps. We'll go very slow, starting at lower current intensities. So there have been publications before showing presence of giant somatosensory evapotentials in patients with increased cortical excitability, different pathologic conditions such as Creutzfeldt-Jakob disease or myoclonic epilepsy. And we showed the same pattern in some of our patients who presented with brain tumors and seizures. And besides having giant SSEPs on averaged SSEPs, as we are typically recording, we also, in these patients who had increased excitability, we noticed these runs of unaveraged SSEPs that kind of stood out from the electrocorticographic recordings during periods of flats. And surely in this patient, we were expecting low thresholds, and indeed we recorded really high amplitude motor evapotentials triggered by stimulation of M1 at very low thresholds. Similarly, we do have seen the same pattern in patients with epilepsy, and this is one of those examples. This is a small child in whom with intractable epilepsy, who had initially severe AG, but in whom we could not identify clearly whether M1 was part of the seizure onset zone preoperatively. And so the patient underwent intraoperative recording with electrocorticography pre-resection and actually throughout the resection for identifying the epileptogenic zone, as well as sensory motor mapping and monitoring during resection. And after we identified a central sulcus with a phase reversal technique, the strip was placed parallel with the central sulcus, and we stimulated M1 with contact-free the violet strip at 80 milliamps, which is pretty low for a child actually under general anesthesia. And we recorded nice brachyradialis MEPs during resection. At some point, we had a drop in the motor evoked potentials. And of course, the first concern was the fact that maybe corticospinal tract was injured or the strip moved. It turned out that as the surgeon was resecting the area that was mostly active, which was luckily the post-central area, the entire excitability of the paralandic cortex decreased. And while we stopped seeing significant epileptiform activity, the threshold of M1 also increased. And that was the reason for drop in our MEPs. Once we increased the stimulus intensity, we had recovery of the MEPs. This patient had no motor deficit, and the ECOG at the end was pretty quiet post-resection. Last but not least, we looked to see whether we could continuously monitor and simultaneously monitor somatosensory evoked potentials from median and posterior tibial SCCPs concomitantly with motor evoked potentials in brain tumor surgery. And what we notice is this cycling and variation of amplitude of the triggered MEP with both higher amplitude and so like excitatory inhibitor, some excitatory inhibitory phenomenon. And this goes back really to the paralandic connectome and to the thalamocortical projections, both postcentrally and precentrally, and to modulation of the cortical connections between S1 and M1. And it's a very similar phenomenon of excitation and inhibition at the cortical level as previously described with TMS studies, but this time, to my knowledge, for the first time, we were able to show that similar phenomena happens also under general anesthesia. And that is in a nutshell what we are dealing with in the OR during these mapping procedures.
Thank you so much. Great presentation. Thank you. We move to the next speaker. Dr. Moldovan has an interesting title, asking himself if it's to burst or not to burst in comatose patients, an EEG story.
Greetings. As it is hinted from my title, I'm talking to you from Copenhagen region, Denmark. But the story today is mainly focused on my continuing collaboration with my alma mater, the University of Medicine and Pharmacy Carol Davila, Bucharest, Romania, which is this beautiful picture here. And as my disclosure, I want to mention from the start, as the past president of the National Neuroscience Society of Romania, I'm going to heavily feature my Romanian collaborators in my talk. And as I used to say, a neuroscientist can walk on water. And I say this very confidently when I think at the state of the water as ice. And brain states have been I was very interested in brain states for a lot of my research career, and starting with my first experiment in rodents, where very simply and minimally invasively, one can record the EEG signals. And in this picture, I'm featuring one of how I was doing the EEG recordings in '92 in the laboratory of Professor Leon Zgrand. And he has, you can see what you can see in this picture is EEG recordings from the electrocortical graphic recordings in a rat in 4 channels here during anesthesia here, and you can see, and then during ischemia, the EEG state changes from something oscillatory to something very flat. In a modern day, you can do the same type of recording, and if you can look at the low level of anesthesia, the EEG is like a full pattern here. We're looking at the 30 seconds of ECG during isoflurane anesthesia. With the depth of anesthesia, EEG becomes discontinuous. As you can see here, there are some discontinuities, and with further increase in anesthesia, EEG becomes like fragmented in some bursts and some suppressions, as you could see in my previous in the previous speaker's talk. And that we call this pattern as a burst suppression pattern. So like my eyes in my slide previously, this is the brain electrical state I was mostly interested in the burst suppression state. And why I was interested in this, because studies done with Dr. Andrei Lie at the time were looking at very short brain ischemias, experimental ischemias, where you could see in the picture here, in the first slide here, you can see the Doppler monitoring of the blood flow, which goes to nearly 0 here, and the EEG becomes quickly flat. But then upon recovery, the EEG recovers backwards through the same sequence. First is this discontinuous burst suppression pattern, and then slowly to continuous EEG. And this is a non-injuring type of lesion. It's a 1-minute global ischemia in a rodent, which has been shown extensively histologically that causes no injury or no deficit. So burst suppression is not necessarily an alarm sign here. And importantly, how you measure this, we have a parameter called burst suppression ratio, which basically says how much time amount of refraction is spent in suppression. And this goes, of course, from 100% when it's fully suppressed to 0 when the EEG is continuous. And at that time, we said, well, can we use this for some kind of a prognostication? And then we had the pleasure to meet in Bucharest Professor Florin Amzica, who came and gave a talk when he was trying to do experiments, recording from both intraneuronally and EEG at the same time, this burst suppression activity. And then he said, well, this would be what we all know to be a spontaneous burst suppression activity. But then when he started stimulating with various stimuli here, including photic stimuli, auditory stimuli, he could evoke bursts for every stimulus. And these evoked bursts were indistinguishable from what we previously called spontaneous bursts. Okay, then, so burst suppression ratio would then depend on how much noise is in the room. That was very interesting idea, or what kind of stimulation there is. So this kind of, is this a big confounder? We were very intrigued by this. And then I had the pleasure to start collaborating with Dr. Mirela Simon here. And then we looked at some patients which underwent burst suppression during surgery. So they were back surgery, so they were normal, and then we could in the reactivity. So you could see here during anesthesia, we then devised such a system here in which we looked at the time before the stimulation, during the stimulation, and after the stimulation, and we could see here that as compared to the burst suppression ratio between the stimulation. During stimulation, in that case was electrical stimulation of a peripheral nerve, the burst suppression ratio was decreasing, and then after, when this was stopped, it was increasing. So we could indeed, the stimulation would cause increased bursting in a human. And the other thing that we thought is that, well, this kind of the ability to induce burst, which is called reactivity, it kind of depends on how much bursting activity was before. So with the deeper anesthesia levels, this reactivity appeared to be bigger. And then I met with another, I had the opportunity and worked with another Romanian colleague, Dr. Dragoș Niță, who was at the time looking at comatose children with acquired brain injury, in which we did exactly the same. So stimulation, it was photic stimulation in this case, and stimulation would also induce bursts, which were indistinguishable from the previous, but with a decrease in suppression there. And what we then said, well, but then if it's the same, that there is a relationship of this reactivity increases with the depths of coma in that case, can we then normalize it? And we have looked at this normalized ratio, so the ratio which is where the actual change in burst suppression ratio was normalized over the burst suppression ratio before, was a better predictive value for the outcome in this cases. Then, okay, but there was a few patients, then we have to use it for something more. Then I come back to my collaboration with Professor Ana Maria Zegran, who is a FENS chat member who facilitated this, this webinar today, and I thank you her for that. And we have a paper together where we look at a model where we try to do just that. So we have modified our rat to look at the visual stimulation, intermittent visual stimulation. And because the rat is somewhat different than humans in the sense that the visual evoked potentials, they are mainly contralateral to the stimulation. Then to avoid this confounder, when we look at bursting activity, we have focused only on recording from the ipsilateral channel when there are no evoked potentials here, as you can see. And then we did the same paradigm that we have previously developed in the patients. And we identified the bursts. We looked at the change, as you could see here. This was a bursting before photic stimulation. This is during, and which was this increase in bursting. And exactly like in the humans, we thought that, well, but then we can normalize it. So normalize it with this change in burst suppression ratio with the burst suppression ratio before the stimulation. And then we look at this in a model of global cerebral ischemia, and we look at 3 days after global cerebral ischemia. And here, as you could see here, we had after the 5 minutes global cerebral ischemia in a rat, which is a model of cardiac arrest, it's injuring. It causes small injury to the brain. And that injury we could measure as a decreased in reactivity, in this normalized reactivity at 3 days. So in a way, this is kind of like, okay, now we have an animal model that we can go further and study these findings that we noticed in those few patients. And then we said, what can we use it for? So one way to look at it, we could use it in neuroprotection study. Now we have a quick measure to do this reactivity studies for neuroprotection studies, and this is a model introduced in our lab in Romania by Dr. Ana Maria Panaitescu, and looking at the perinatal asphyxia model, which is, and then we tested the neuroprotective effect of oxytocin, and we showed that there is a decrease in burst suppression ratio, which can be rescued partially by neuroxytocins. And now Dr. Vlad Morozan is now continuing these neuroprotection studies. But now everything we spoke now is about decreasing, reactivity as a sign, but can the reactivity increase? And another PhD student in our group, Dr. Lorenzo Tofan, is now looking exactly at that using an epilepsy rat model of absence epilepsy, and where we can do the same type of measurements when we show that we can find an increase standardized burst suppression reactivity in these rats, and this is sensitive to anti-epileptic treatments. But okay, now very quickly in the last minute here, I would like to talk about human studies, because for us now, okay, we can do this in an animal, but there are not so many patients in birth suppression. Can we do kind of similar type of reactivity measures in patients with continuous EEG? And here we start talking looking at the human patients together with Joanna Mandruza, who is a collaborator here, and we developed a method which we call default EEG macrostate reactivity, which uses the same principle. It takes a multi-channel continuous EEG here, which can then be converted to a spectral matrix, which can be clustered in different classes, which we call macrostate classes. And again, a binary signal can be extracted like the birth suppression binary signal, but now this can be extracted from the continuous EEG. And this is done together with Dr. Cosmin Sherban, who is an engineer, and he has really worked hard to implement this at the level of several patents. And what can this be used for? We can look at the measure of awareness, and this has been a study recently published in the recent FENS spatial issue, where we look at this changes in this default EEG reactivity in relation to consciousness, together with Dr. Mihai Stancu. And then very last, now, okay, we can measure this reactivity in continuous EEG with a default EEG reactivity. We can measure the birth suppression ratio in birth suppression. But now we have shown for the first time, and in this patient, that we can use basically the default EEG reactivity across the whole spectrum. So now we have one index which can be used for both continuous and discontinuous EEG. Thank you very much for your attention.
Thank you, Mihai. Very interesting presentation. And in the meantime, I have to remember you to pose questions, make comments, and we will be very happy to answer to you very soon. So the last presentation of our webinar will be coming from Dr. Irina Oane from the University Hospital of Bucharest, my colleague in my group.
Hello, thank you for the invitation. So in the next minutes, I will try to show you how we use the direct electrical stimulation in patients that undergo epilepsy surgery, pre-invasive exploration of intracranial electrodes to study the cortical excitability. So these are my disclosures. So stereo EEG is a method that is internationally used to determine the location of the epileptogenic zone and to map functional cortex. And this implies that the patient undergoes implantation with several electrodes in structures that are determined pre-operatively. And it's the main advantage is that it provides access to virtually any region of the brain. And the second advantage is that this is a chronic exploration. So that means that the patient stays with us in the EEG unit for several days, up to 2 weeks, so that it gives us access to perform anatomic electroclinical correlation. So what are the mechanisms of electrical cortical stimulation in this setup? So practically we inject an electrical charge in the extracellular space that will have two results. So it will have a passive spread and a synaptic spread. And we are more interested in the synaptic spread, which is due to the depolarization of the initial part of the segment of the neuron. and we create a non-physiologic activation and antidromic activation. So the clinical effect of this stimulation could be excitatory. So, for example, if we stimulate the primary cortices like motor or visual, we will get hallucinations or movements of the hand or visual hallucination. But if we stimulate associative cortices, like multimodal cortices, for example, perisylvian structures or the language network or parietal cortices, we will get inhibitory effects. So practically, we will block a function of the brain. If we stimulate while the patient is talking, we will block the speech, or if we stimulate while the patient is counting, we can get this calculation. So there are several protocols that we use in the clinical setup. They have some common features. All the stimulations are performed bipolar, and we use a biphasic stimulation. And usually the pulse width is 1 millisecond, or it can be 3 milliseconds. So with this type of stimulus, we have different types of applying the stimulus. For example, we have the single pulse electrical stimulation, in which we apply 20 pulses. The interstimulus interval is 15 seconds, and we vary the intensity of the current from 0 to 5 milliamps. And this type of stimulation is usually used to map effective connectivity. In the next protocol, we use low frequency stimulation. So we deliver 1 pulse every second for 40 seconds, and we vary the current from 1 to 3 milliamps. And this is usually used in clinical practice for mapping the epileptogenic cortex, because we know that this type of protocol is at the high sensitivity and specificity for inducing seizures. And of course, if we are delivering this stimulation protocol in primary cortex, we can also get functional responses like clonias, or we can also map visual cortex. And last but not least, we have the high frequency stimulation protocol, which implies delivering 50 pulses every second for 5 seconds. And this type of protocol is usually used when we want to map functional cortex. So, irrespective of the type of protocol we use, what we are getting from an electrophysiological point of view is a corticocortical evoked potential, and this is how we can measure excitability of the brain. So there are several ways, several methods to analyze, to perform this analysis. So in the first situation here, you have the way in which we analyze the single pulse electrical stimulation. So what we get when we apply stimulus is, as I mentioned, a corticocortical evoked potential. And by measuring the root mean square of each evoked potential, we can get the excitability of the area that recorded the stimulation. On the other hand, there are other types of measuring this evoked potential. So there are some groups that measure the properties of the evoked potential, like peak latency, onset of latency, which are usually showing the dynamic of the response. And also the strength of the response, for the excitability is measured by the amplitude of the response. And by computing, by analyzing this excitability, we can also get connectivity, effective connectivity of the stimulated area. So what is more interesting now is the fact that we can also analyze this the excitability, brain excitability, and connectivity in relation to high frequency stimulation that is inducing the functional response, a clinical response. And this is we are able to do this because we are using a specific type of stimulation protocol, which involves alternating the polarity of the pulses. So by alternating the polarity of the pulses, we can disentangle the response of the tissue, which is nonlinear, from the linear response, which is artifactual. So when we analyze this type of stimulation, we can apply time domain analysis or frequency domain analysis. And here you can see in the frequency domain analysis how we get the artifact and how we can analyze the responses that are within the frequency of the stimulation. So in the upper part, it is an example of how we can correlate cortical excitability and function. So by subtracting the amplitude of the response of the stimulation that induced the clinical response from the baseline, we get the regions with the responses which are time locked to the stimulation. There are also other ways in which we can analyze this type of stimulation in this complex space, which allows us to perform phase locking analysis, which is the connectivity of brain and body. So now I will show you some examples from our clinical work. So for example, we can perform this type of analysis in patient level. So here we have a stimulation, so several stimulation in which we have induced clinical responses, somatosensory responses, and we get the cortical areas with a significant response. We can analyze this response, or we can display this response using connectivity metrics or circular diagrams, but from a clinical point of view, I think that the 3D representation is it's the most suitable. We can see that this area that was stimulated is connected with the premotor cortex and with the parietal operculum. So on this slide, we have a measure of cortical excitability of the areas that are stimulated, so the threshold map. In which you can see here color code. So 0 is a very low stimulation threshold, and 3 is a high stimulation, high threshold. And you can see that in our cohort of 27 patients, the primary and the premotor cortex, pericillian cortex, and occipital and temporobasal have the lowest threshold, and there are also areas in which we could not identify a clinical effect. So high areas, high threshold areas, which is the prefrontal cortex. And then on the other side, we have the response map. So areas that got a response during the stimulation, and they follow the same pattern of activation. So the same type of analysis we can use in order to map excitability of areas involving language. So here with the green dots, you can see areas in which we induce the language responses during chronic stimulation and the response map associated with these stimulations. So what is even more interesting is the fact that we can combine several types of analysis measuring cortical excitability and connectivity. And here it is an example in a patient in which a very particular clinical effect was induced, so autoscopic hallucination. And we can map cortical areas activated time locked to the stimulation that induced the clinical effect. The same almost the same areas that are activated during resting state using the cortical evoked potential during single pulse electrical stimulation. And type of connectivity analysis, which is a spontaneous type of analysis, which could be two ways. So it shows areas that decrease in connectivity and areas that increase in connectivity when we compare stimulations that induce the clinical effect with stimulations that did not have clinical effect. So we can couple this functional information with structural connectivity that we subtract from diffusion weighted imaging on pain-specific patients MRI. So, and last but not least, we can apply this type of excitability and connectivity analysis to map a particular region of the brain. And here is an example of how we have mapped the connectivity of the cingulate cortex. So all this is available. Available in this atlas, where you can see different types of connectivity measures, either structural or effective of each brain area. Mm-hmm. So thank you very much for your attention, and thank you very much to the Guggenheim Parkville and to the authorities that have funded our research.
Thank you, Irina. We are close to the hour that we need to end our webinar, but there is still some time for questions or comments. There is a participant, Mariana Carrera, asking Mirella Simon, how do you reset a certain brain region during surgery?
So I don't think I quite understand the question, unfortunately. What is meant by reset? What was meant by reset?
Well, this is the question. Maybe you can comment on this.
I mean, depends. Depends what the question is. So I'm not sure what reset means.
Well,
we
maybe
don't.
you can change the excitability of a region during surgery in a certain condition.
Oh, well, we can definitely influence the excitability depending upon the particular conditions for that patient. For example, if I have a too deep burst suppression, I may talk to the anesthesiologist to, decrease the propofol. Even simple irrigation of the cortical surface with ice cold saline that we use frequently to abort after discharges during the stimulation influences, let's say the M1 threshold. So we can have increase in M1 threshold. So it's really some situations we can't do much about it. I mean, if I know I have a patient with increased excitability, there is this balance, right? You have low threshold for depolarization, that's why mapping M1, but also you have a low threshold for after discharges. So it's this balance that you got to keep between, you don't want to continue CG. I would never aim actually to map a patient who presented with seizures on a continuous EEG under general anesthesia. Actually, I prefer to have a little bit of burst suppression because that's very protective for this after discharges. Even if I trigger after discharges, the organization and hypersynchrony and propagation will be stopped by presence of a discontinuous EEG. So there is always this balance. How much suppression do you want, and how, where your threshold is. For children, for example, where we have, we struggle sometimes to reach the M1 threshold, then definitely I would, try my best to persuade the anesthesiologist not to use inhalation agents, because that would be the end of, our ability to map, or be very difficult to map, but rather to use, TEVA. So there are certain things that we can work with, and that pertains particularly to anesthetics, and maybe be aware of what else is happening in the field, like irrigation, and so on. Antiepileptics also, we actually administer load patients with antiepileptics before the mapping procedure, regardless if the patient has epilepsy or not. And we looked into it. It's actually very protective, for triggering seizures during the mapping. And we also looked to see whether administering these antiepileptics influences the threshold for mapping, and in our population it didn't, which is kind of intuitive.
Yeah.
You shouldn't, so.
Thank you so much. I guess you gave enough details. We have another questions coming from Nina Dorn for Professor Moldovan. In some comatose patients after hypoxic ischemic injury, burst suppression EEG is very identical and synchronous. Burst is a clear sign of poor neurological outcome. Nevertheless, with only a minor stimulus, clapping, arm stroking, a big burst can be induced in the entire brain. In this case, the reactivity probability, as defined by Professor Gaspar, would be very high. Am I correct? So this would suggest a relatively intact brain, while we know this patient will likely not recover, suggesting a highly damaged brain. Would this be corrected by the normalized reactivity measure of Professor Moldovan? And would including the complexity of the evoked response have any added value for prognosis? For example, with a perturbational complexity index?
Thank you for the question. Yes, I mean, to start with, we do not have prognostic value for this normalized index. But we really hope it's going to cover both by normalization. It should give a better idea for both increasing or decreasing reactivity as compared to a baseline. For us, at least in the animal studies, normalization helps a lot to diminish this confounders. So yes, I mean, abnormal reactivity can be both reduced reactivity and increased reactivity. In both cases would be abnormal, as therefore in both cases would be a bad prognostic value if it's if it can be measured. So yes, that's a very good point.
Thank you. We have a question from Violeta Caraja for the last speaker, for Irina Oane. Regarding high stimulation strategies in controlling epileptic seizures. To your knowledge, did, for instance, cerebellar stimulation work in humans for inhibiting temporal lobe seizures, like it was shown in rodent studies? And there is a link to one of these studies.
Okay, so thank you for the
question.
So there are
several types of stimulations that have been tried in humans for controlling seizures, not particularly temporal seizures. So we have vagal nerve stimulation or types of vagal nerve stimulation, but we also have stimulation of the epileptogenic zone with the responsive neural stimulation. But as far as I know, there are no clinical applications of stimulation of the cerebellum yet. We are looking forward to it.
Well, thank you so much for all your interesting presentations. Thank you for your questions and also very nice comments that are underlying the interesting aspects of our session and also proposing a future collaboration. I thank to the organizers for the intention, for the idea, and for the whole support. I thank Professor Zagran for her initiative, and I wish you a very pleasant day and evening after. Thank you.